Hello friends, this is the video on the citric acid cycle, and you can also call it Krebs cycle or tricarboxylic acid cycle. In eukaryotes, this TCA cycle takes place in the mitochondrial matrix, and in prokaryotes, this TCA cycle takes place in the cytosol. So now let's go to the detailed mechanism, the pathway of this citric acid cycle. This is the acetyl-CoA and the oxaloacetate, which are the key component of this citric acid cycle, and the first two reactant. This is a 2 carbon molecule and this is a 4 carbon molecule, which combines and form this citrate of a 6 carbon molecule, and this is the first stable molecule of this cycle. So according to the name of the molecule, this cycle has been called as citric acid cycle. So now, from where this acetyl-CoA has come from? This acetyl-CoA is had been produced from the pyruvate by the pyruvate dehydrogenase complex, and this acetyl-CoA enters into the mitochondrial matrix where a smaller amount of oxaloacetate is present. Look at the structure, and they combine together by the enzyme called citrate synthase. This name means that they are the enzyme which is actually responsible for combining two different molecules. And here is the structure where this double bond in between the carbon and oxygen breaks, and one hand is open, which actually captures this carbon of the acetyl-CoA and forms this structure. So if you look at the structures, you will be able to understand which hand of the carbon is joining with which carbon and what. So it will be easy for you to understand what reaction is going on. And this acetyl-CoA, the carbon that has been donated by the acetyl-CoA, is in the pink shade. Look at this one. Yes, this one. This is the carbon that has been donated from the acetyl-CoA. So now this citrate is a 6 carbon structure. It undergoes a dehydration reaction by the enzyme called aconitase. This aconitase takes out the water molecule from the structure, and now you have to understand which hydroxyl group and which hydrogen has been taken out to form this water molecule. Look at this structure. There is one hydroxyl group and there are two hydrogen. One of a hydrogen has been taken out to form one molecule of water, and so after taking out this hydroxyl group and one hydrogen from this carbon, two hands are free of these two adjacent carbons, so they have formed a double bond in between them, and this is the structure. Now this structure is called a cis-aconited. Now again a dehydration step is there, so one molecule of water is again added, and now this time this hydroxyl group has been added to the last carbon. I mean the second this double bond has been the double bond is in between these two carbon, these two adjacent carbon. After this hydrolysis, this double bond breaks and the hydroxyl group is attached to the second carbon of the two, and the first one gets the hydrogen. So if you look at this structure and this structure, you will be able to understand that the hydroxyl group has only changed its position from this carbon to that carbon, and this new structure is called the isocitrate. Now this enzyme, this reaction from the production of the isocitrate from the cis-aconitate is again accomplished by the enzyme called aconitase. Now the next step is oxidative decarboxylation. Decarboxylation, from the name itself you can understand that one carbon molecule has been taken out from the structure, and this is the carbon molecule that has been no, not this one. This is the carbon molecule that has been taken out from the structure, and the enzyme responsible here is the isocitrate dehydrogenase, And you must be aware of one more thing, that is the enzyme dehydrogenase means that NAD plus is working as a cofactor, and probably one hydrogen has been taken out from the substrate to make NAD plus into a reduced form, that is NADH plus H plus, which will again go to the electron transport chain and will produce ATP in the mitochondria. So this is the structure of the isocitrate, and this is the COO minus group, which will be out in the form of carbon dioxide, and the hydrogen that has been taken out is this hydrogen and this hydrogen. So there is so after losing its hydrogen, this carbon is left with an oxygen molecule, and double bond is in between them. And where this hydrogen has gone now, this hydrogen has now formed NADH plus NADH by joining the NAD plus, which is working as a cofactor with this isocitrate dehydrogenase. So this step is a bit complicated, and in this step one carbon dioxide molecule has been taken out from this carboxyl group, as this carboxyl group is leaving this big structure, and at the same time one hydrogen has been taken out, which will join the NAD plus, which is working as a cofactor with this enzyme and form the NADH, and the resulting molecule is the alpha ketoglutarate. Okay, alpha ketoglutarate. Now again this two carbons are from the acetyl CoA, and this is the rest of the structure. Okay, now in the next step, which is again another oxidative decarboxylation, so again another carbon dioxide will be produced and one carboxyl group will leave, and this is the carboxyl group which will leave in the form of the carbon dioxide. And here this alpha ketoglutarate dehydrogenase complex now will produce again the NADH plus H plus from the NAD plus, and which what will happen here in this structure is this, that one coenzyme A will be added to this hydrogen, as this COO minus group is leaving. So this hand is now open of this carbon, and this hand will be replaced with the coenzyme A, and one molecule of NADH plus H plus will be produced, and this structure is called a succinyl CoA. Alright, so now there is a next step, which is a substrate level phosphorylation, and it's a very important step. Here the enzyme succinyl CoA synthetase is working. So now what is happening here, that this coenzyme A will leave, here is the structure. So and the new molecule that is produced from the succinyl CoA is the succinate. So look at the structure, and succinate is a symmetric molecule, because if you number the carbon from this side or from the opposite side, it will be same. So it has got a symmetry. Now if you compare these two structure, you will be able to understand that that in this last carbon, this coenzyme A was there, it has now left, and it is now replaced with this carboxyl group. And what is this substrate level phosphorylation? Actually this substrate level phosphorylation is this, that there is no phosphate in with this substrate, which can be used to produce GDP, to produce GTP from GDP or ATP from ADP. Rather, an inorganic phosphate from the outside comes into play, and during this reaction it forms intermediate form by joining this substrate. This phosphate and this substrate forms an a combined molecule which is of a very short living molecule, and with the help of this enzyme, this phosphate group joins this ADP or GDP to form GTP or ATP. So now if you want to get a clear idea about that, I would like to show you my next slide. Sorry, this is my next slide where I have shown the substrate level phosphorylation details. Here in this step, this is the succinyl CoA, and this is the inorganic phosphate that comes from outside. It is not of the this is not the part of this substrate, and here is the enzyme, that is succinyl CoA synthetase, and this histidine is in the active core of this enzyme. What happens that this is a phosphate group, a inorganic phosphate, which is a very strong nucleophilic factor which will attack this structure. Look at this structure. It will attack this carbon, and so this coenzyme A will leave, and this phosphate group will join and will form a very complicated structure, and this structure is a very short living structure. Okay, and now after this coenzyme A has left, this histidine residue of the enzyme core, active core, will come into play and will attack this phosphate group and will break the bond in between the succinate molecule and the phosphate group. The succinate will leave, and now it is attached with the histidine. Now histidine will introduce NDP. NDP is actually the ADP or the GDP. Look at this place, and it will transfer this phosphate group on the NDP and form this NTP. This NTP can be ATP or GTP, and then again the enzyme will come into its original form. So in case of substrate level phosphorylation, no phosphate group was previously there in this substrate. Rather, an inorganic phosphate has been added from outside, and it will form a very transient structure with this substrate, and then it will make some change into the substrate and will form a new product. Here this succinyl CoA has formed this succinate, and then this enzyme will add the phosphate group to NDPs to form NTP and will come back to its original form. So now again, if you go back to our previous slide, this is the same thing that has happened here, that this we were just talking about this step only, and this coenzyme A has left, and now look at this structure. This is the structure. This is the carbon now is with the hydrogen, and it is this carbon that is with the hydrogen, and the last carbon that which was attached with the coenzyme A, now it is in the form of a carboxyl group, and one GTP or ATP has been produced. So now this succinate, which is a symmetric molecule, undergoes dehydrogenation and will produce fumarate. Dehydrogenation means one hydrogen will be removed from this structure, and the cofactor which is working along with this enzyme called succinate dehydrogenase will produce FADH2. So the FAD plus will join the hydrogen and will produce FADH2. It will also go to the ETC, electron transport chain, in the mitochondria, and will produce ATP. Sorry. So now this succinate undergoes the dehydrogenation. So which what is really happening? This carbon has two hydrogen and this carbon has two hydrogen. Both of them will leave one hydrogen molecule from each carbon, and so they will form a double bond in between them, and these two hydrogen molecule will join the FADH2. Alright, now there is a hydration step under the control of the enzyme called fumarase. This double bond will be hydrolyzed, and look at this structure. This is a malate, this is a new product from the fumarate, where this double bond is broken and this second carbon has got a hydroxyl group and the third carbon has got the hydrogen group. Now the next step is a dehydrogenation step. So dehydrogenation means one hydrogen will be taken out from the substrate and will produce NADH plus plus H plus or FADH2. Here in this case, this malate dehydrogenase, this enzyme that is working, it has got NAD plus as a cofactor. So NADH plus H plus molecule will be produced, and which hydrogen has been taken out? This hydrogen has been taken out. So now the oxygen now this now a double bond has been produced in between this carbon and oxygen. So again now we have produced this oxaloacetate. And why this is a cycle? Because we started from oxaloacetate, again we have produced this oxaloacetate at the end of this cycle, and this oxaloacetate will now again join the acetyl CoA, which has been produced from the pyruvate, and this pyruvate had been produced by the metabolism of glucose by the process of glycolysis, and these two will join and will form the citrate. Again this citric acid cycle will continue. So and this one I have already talked about, the substrate level phosphorylation. And now at the end of this video, if you want to see some more videos, you can visit my website, which is easyscience.weebly.com, and I will also upload this video in the YouTube, and you will also be able to see this thing. And if you like my video, you please subscribe my channel, and you can also send me mails at mukhop, MUKHOP. I'm writing this thing. Sorry, I want to change the color. This is my email address is MUKHOP, mukhop89 @gmail.com. Here you can send me your feedback, suggestions, or if you have any kind of a question. Thank you for watching my video. Stay tuned for more.